Okay, I think we are good to go for today's class. And I would like to welcome you to the sixth round of the Ecosystem Modeling course at UBC, FISH 501, a graduate course. And today we are going to have a guest lecture by Carl Walters, who'll be talking about the foraging arena and things to watch out for when you're developing EWE models. So thank you and welcome to UBC. Which is actually what you see in the picture here, including the student housing here. Can you imagine these poor students that are forced to look out at a landscape like this? It must be tough. Anyway, UBC is situated in Coast Salish territory, and we acknowledge that we are at the traditional, ancestral, unceded territories of the Musqueam, Tsleil-Waututh, and Squamish people. And I also acknowledge that we have participants from many countries, and I welcome you, and I'm very, very happy that we, thanks to COVID, has the opportunity to include people from outside of UBC. That's a privilege, and we very much appreciate that your interest in being here. I'm happy to introduce Carl Walters. OBC is Order of British Columbia, and that's the highest order you can get in British Columbia, and it's hardly ever given to a scientist. It was given to Carl as a recognition of what he has meant for BC. He has trained several generations of scientists, lots of them fishery scientists working in fisheries and ecology and many other fields, and he's been here for, he'll probably tell us about some of that story in a little while. But Carl yesterday gave a talk in which he paid tribute to Ray Hilborn, who was recruited here shortly after Carl came as a postdoc, as a PhD student, and he called Ray #1 fishery scientist in the world. I think Carl was being a bit shy on that one, modest on that one. I put Carl Walters as #1 and Ray only as #2. That's up for discussion, of course, but Carl has been very instrumental in this, and this has been recognized by American Fisheries Society, the Award of Excellence by ICES in the Prix d'Excellence, whatever it's called in France, and also, of course, by the Volvo Prize and many others. His research spans very widely. If you think about adaptive management, or some call it now active adaptive management, well, that was described and developed by Carl and Ray and Buzz Holling way back there in the 70s, and described by Carl early on. He's been really instrumental in that whole concept. Management strategy evaluation comes from there, and I could mention ecosystem modeling, and you'll hear more about that in Carl's lecture. So I'm not really going to say much more about him, because sometimes a picture says more than 1,000 words. So rather, I am going to pass on and stop sharing my screen and ask Carl to share his. Carl, welcome, and thank you very much for giving the lecture today. Oh, and the lecture on Tuesday as well.
Okay. So I'm going to give you a pretty wide-ranging talk today that will primarily talk about why we've been able to develop ecosystem models using Ecosim that seem to work fairly well. And I'm going to talk about a lot of biology and things like that along the way. The answer to the question that you see in the title there is yes, we can, as you've already heard from other speakers. And I'm going to basically explain to you why we can do that. And all of it comes because of a lucky accident that I'll describe for you near the start of the talk. But let me begin here by talking a little bit about the history of ecosystem modeling. because I was involved in pretty much that entire history starting in the 1960s. Billy has mentioned to you that ecosystem modeling began in earnest in the late 1960s during the International Biological Program. That program was the first time that teams of scientists, ecologists and physical scientists and so on got together with the agreement to study ecosystems, particular ecosystems around the world. And the reason, one of the key reasons they were willing to do that was that there was a promise in the IBP from people who were starting to use systems modeling techniques that they would be able to synthesize the component research studies in these projects into ecosystem models that would be useful for making management predictions and things like that. Now, starting about 1970, as results started to come in broadly from these IBP studies, a lot of the projects realized that they didn't have the capability to do their own modeling. They didn't have mathematicians and so on working on the projects. So they, because I'd been publishing a bit of modeling work on a couple of the IBP projects, they came to us at UBC and they asked if I was willing to go around to modeling, to IBP projects around the world and help them start to develop models. And about that time, I happened to have a couple of really, really bright graduate students, particularly Ray Hilborn, Randall Peterman, who recently retired from SFU, my wife, Sandra Buckingham, who were quick at developing computer programs. They were quick programmers. And I think it was Ray came up with this idea. He said, well, look, if we really want these people to, these IBP people we're working with to understand what we're doing and to appreciate the need for the modeling and so on, what we really need to do is to sit down with them and actually build the models. The people who learn the most from modeling are always the people who do the modeling, not those who later perhaps use the modeling results. So Ray came up with this idea that we could go someplace where there was an IBP project or have their science team come to us, and we could sit down and in a, say, a 5-day workshop period, we could talk through a model structure with them. And then the graduate student team could actually build the model on the computer and get it running and get it into a point where these people from the IBP project could play with it and understand its limitations and see what gaps of information and so on might be present from difficulties in providing parameter values for such models. So we developed this workshop technology, and we used it on a whole bunch of different projects, and we started to broaden it out with support from Buzz Halling to looking at a variety of management problems. But the basic idea was always that you have this little team of poorly paid grad students that are willing to work all night long for 5 days and build these models. And it turned out to have quite an impact on these projects. One of the main impacts that it had was that most of the modeling efforts were essentially unsuccessful. And the reason for that is that people had not realized when they were designing the projects what specific relationships and observations and data they should be collecting. So we had a series of people from different disciplines collecting data that would be appropriate for their discipline, but not for building an ecosystem model or synthesizing information. So one of the first big lessons from this was if you're going to have a big project and get a lot of people together, start out by building your model. Don't expect somebody to be able to take all the random data that you collect afterward and put it into a modeling framework, because the data are not going to fit together. There'll be huge gaps and missing bits of information that nobody happened to think of at the time when they were designing their field projects. So the modeling workshop idea came to be thought of as a way of organizing research projects at the outset as much as to try to bring together data afterward. No, but there were a couple of IBP projects. One in particular called the Marion Lake Project that was in a little lake up the Fraser Valley from Vancouver near Mission, where they had a large team, actually 25 graduate students at one time during the project, out there studying everything they could think of in that lake. And so they were monitoring abundances of something like 800 or 900 different species of organisms, ranging from a wide variety of aquatic insects and other aquatic invertebrates through to fish of a couple of different kinds. And so we thought, well, you know, our models, we're getting quick at this. We're getting good at this. Let's try to build more and more realistic models of that Marion Lake ecosystem that include more and more of the biological detail, more and more of the species that are being monitored, and more of the trophic interactions. And let's use best available theory from people like Buzz Halling in order to predict trophic interaction relationships using functional response models and other modeling tools that were emerging at that same time. Well, we were horrified to discover that the bigger and better we thought these models were, when we built one model that had 800 species in it, for example, the bigger and better that we made the models, the more and more those models misbehaved. The predators would come along and eat up all different kinds of prey. Competitive interactions would eliminate a large proportion of the organisms through competitive interactions. The models were violently unstable dynamically over time, and they could never get. The better we thought the model was from a biology point of view, the more poorly it behaved in the mathematical modeling. And that actually led a bunch of us, including me, to give up on ecosystem modeling, to say there's something going on out there that we don't understand. There's some fundamental property of these ecosystems that we are not putting in these models. And we don't know what that is, but let's just give up and go back to studying simple things like population dynamics and adaptive management, that kind of thing. And so it went for about 20 years. And then this happened. I was out fishing with my son, Will, who will be talking to you. Will was about 7 years old, and we were sitting out in the middle of a lake called Roach Lake up in the BC interior. And Will was leaning over the side of the boat, and he was looking down in the water, and he could see these large zooplankton organisms called Daphnia cladocerans in the water. And we weren't catching any fish, and he was bored. So he's watching these little creatures swim around. And he says, Dad, he says, how come the fish don't eat them all? These things are obviously really tasty looking, and they're easy to catch. Why aren't the fish out here eating them? And then we could catch the fish. So I just looked at him and said, well, those Daphnia aren't attractive to the larger fish that you and I are trying to catch. They're attractive to juvenile fish, but the juvenile trout are not out here in the middle of the lake where we're sitting, because there's really high predation risk out there. A small trout coming out, a Daphne eating trout that came out in the middle of the lake like this has a lifespan of about 15 minutes before a large trout will come up and grab it and cannibalize it. And I said, so, well, all those juvenile trout are over against the shoreline. They're hunkered up in shallow water in weed beds and so on, in places, refuge places where these big fish can't get at them. And then they only come out and feed right near shore for short periods of time each day. And so most of these Daphnia you're looking at aren't available to the major predator that might eat them. So Will nodded his head and said yes. And then I was sitting there on the boat thinking, oh, holy Christ, what have I just told him? What have I just said about the organization of ecosystems here? I mean, everybody knows this. that we see highly organized spatial behavior, particularly of small organisms, that's driven basically by fear, by predation risk. So I said, well, Will, draw me a picture of all the places that, in places you've been, like coral reefs in Australia and swam around, and all the places where you've seen the kinds of behavior that you've seen fish exhibit that is of the kind I just described for you. And he says, well, I says, okay, yeah, we got your juvenile trout and they're coming out and they're feeding near the shore. And then there's fish in schools that hide inside the schools so that their sister will get eaten rather than them. And they're foraging right around the edge of the school in a very restricted volume of water. And he knew about fish that have vertical migration, like kokanee salmon, that hide down in the depths of lake water columns most of the day and then come up and forage just at dawn and dusk at the lake surface. And then he knew about things like juvenile cod that hide under the rocks along a rocky slope in the ocean and come out and forage on benthic invertebrates over mud and sand bottoms for brief periods each day. So this kid was able to describe a pattern of spatial organization that we now think is basically universal out there. That most organisms spend most of their time hiding from predators, especially small organisms. They forage in very restricted spatial volumes. Those restricted foraging activities in these things we started to call foraging arenas actually create two limitation things. One of them is they create intense competition among the juveniles in these restricted areas where the juveniles feed. They also mean that most of the food organisms that I would see if I went out there and towed a plankton net across this lake are not vulnerable to those juveniles, right? And the juveniles are not vulnerable except for a short time to their predators. This is the idea we call it takes three to tango. That one behavior creates two foraging arena structures. One restricted access of predators to the juveniles, and second, restricted access of juveniles to their food supplies. And there's an exchange process back and forth with food in and out of the places where these juveniles feed, and of juveniles in and out of the places where predators can feed on them when they do come out from hiding. Whoa. So we started thinking about the implications of what Will had got me to tell him that everybody already knew, but had never been put in anybody's models, right? The models were all built basically using ideas from reaction vat chemistry, that everything's all mixed up out there and everything's interacting with everything else, like as though you'd stirred them all together in a great big pot, called the mass action principle in a lot of modeling. And it just isn't that way. Well, right away, we could see that there was a whole bunch of patterns in population and community dynamics that are explained by this simple proposition that fear drives the evolution of foraging in spatially restricted arenas, in spatially restricted times and places. We see this all the time. We see apparent food limitation in the presence of apparent plenty. We see juvenile trout that grow poorly in lakes, even though those lakes are full of Daphnia, because the juveniles aren't out where the Daphnia is. As I'll tell you in a minute, an implication of this is what's called the Berton-Holt recruitment relationship that's very widely observed in fish population dynamics. We also have seen in a lot of historical data very weak dependence of natural mortality rates of fish, particularly on the abundances of predators. Predators can go up and down a lot, and the natural mortality rates of those fish don't change much at all, even when most of the mortality that we do see is due to predation. Adding more predators doesn't increase mortality rates. We also see a thing called ratio dependence. We see a kind of stabilizing effect in ecosystems of creatures feeding on each other in predator-prey interactions, as though they fed at rates dependent on the ratio of predator-prey abundance, which simple ecological theory says will cause ecosystems to be more stable. Okay. We also see a thing called positive covariation in predator and prey abundances along productivity gradients, which simpler predator-prey models don't predict. They predict that when you go along a productivity gradient, more productive areas should have more predators, but not more prey, because the predators should build up until they drive prey densities down to the same density in all the areas, no matter how productive those areas are. Okay, so we then a little later on, I guess one way of saying all this is the same fine scale variation that drives us crazy when we're out there trying to measure things in the field is also critical to long-term dynamics and of ecosystems and to ecosystem stability. This right here is actually the most important message from this talk. Okay, well, we started thinking about how we could model this idea of spatially restricted foraging and creatures being safe most of the time and invulnerable only a small part of the time. And we said, well, let's just think about it this way. Let's think about a prey population like the zooplankton that those fish are feeding on Billy's picture, as most of them as being invulnerable because they're not in places where the predators are, or if they're predators, they're hiding from their predators. And then they have to come out or are mixed into the area where the foraging is taking place. And they become vulnerable to predation, and they mix back into the invulnerable pool. What rates depend on their behavior and how short a time they spend foraging and so on. And it's just these vulnerable prey that are attacked at rates that are proportional to the vulnerable prey density and to how many predators there are chasing them in these foraging arenas. Okay, well, this it turns out that if you just take this, do a simple mathematical model of this, and then because these exchange rates are going on really fast, the vulnerable prey density will come to equilibrium very quickly with respect to these three rate processes. And when you predict what that equilibrium vulnerable prey density looks like, it should look like it should be positively proportional to how many total prey there are, and it should be inversely proportional to how many predators there are. That the vulnerable prey density should decrease in an inverse pattern with increasing predator abundance. Well, that's a funny looking, strange little equation. Let's just do a graph of that equation. It's the same thing, saying the same thing. The graph of that equation says that as you increase the density of competing, say, juvenile trout in the little areas where they're feeding near shore, that the food density in the places where they're feeding should decrease as what's called a hyperbolic, or 1/x relationship to the number of juveniles that are feeding there. Now, an exercise I used to do with graduate students, I used to draw this graph without any curve shown on it. And then I would ask them, as you increase the number of juvenile fish, how should the food density available to them change? It should go down when there's more juveniles competing for it, right? But what shape should this relationship have? And I can tell you from having given this in class exercises in year after year after year back in the day, is that every student and every scientist that I've asked this question will say, well, it should go down linearly. The more competitors there are, there should be linearly less food available per predator. But this weird foraging arena exchange model says no. Food density should be most severely affected with the first increments in juvenile competitor density, and then not much affected as density increases quite a bit after that. This is what's called in modeling a counterintuitive prediction, where just building a simple mathematical model leads you to a very different prediction than you would intuit if you just tried to draw a graph of what you thought the relationship was going to be like. In fact, this is the best example I know of from all my experience in ecology of a counterintuitive prediction from a model. spectacularly against what everything our intuition would tell us, and also against what we would expect from a lot of simple ecological theory like logistic population growth theory, which does not predict this at all. It predicts straight line decrease in reproductive performance as predator abundances increase. Okay, well, as it happened, there was a group of people, including Eric Parkinson, who some of you may have heard his seminar here last week in our quantitative series, went out and did an experiment on a bunch of small lakes in the interior of BC where they stocked the same little juvenile rainbows that Will was looking at different densities in a whole series of ponds. So they had low to high stocking rates. And then they measured the body weights of those juveniles after a couple of months in the lake. Now that body growth is a pretty good indication of the food densities that the juvenile fish were seeing. And lo and behold, what do they see? No straight line. They see the first increments in stocking density generating a really dramatic drop in growth and body weight, and then further increments in up to really high juvenile stocking densities, not generating much lower growth rates than the seen after the first increments. So exactly the pattern, this counterintuitive pattern predicted by the foraging arena theory equation, which was what we were starting to call it at that time. Now there's an interesting observation right here. There's an outlier from this hyperbolic relationship. It's this point right here. This little point was a lake that was stocked at a very low density in the spring, but the fish exhibited very poor growth. Well, it turns out that what had happened in that pond was that the previous fall they would do they had done gill netting in the pond to remove any fish from their previous treatment, so the pond would be free of other things at the time when they did the stocking. But the pond iced up on them before they could gill net out all of the little fish that had been there from a previous treatment. So at the time when the stocking was made, there was a whole bunch of older rainbow trout, bigger rainbow trout that had overwintered from the previous year's experiment. So when these juveniles were stocked, the first thing those juveniles saw was bigger rainbow trout all around them. And the first thing they did was to move into shore and radically reduce their feeding activity in response to the very high predation risk that these juveniles thought they were going to experience. So what we see here is there's a food effect. There's also a predator density effect in these data. This experiment was then repeated with systematic variation in predator densities and so on to show that yes, indeed, at any juvenile density, as you increase predator densities, you drive, it doesn't take very many predators at all to drive juvenile fish to exhibit dramatically lower growth rates. There's now a comparative literature on what's called the ecology of fear, where people have done experimental stocking of manipulation of predator densities and look to see what it does to performance of their prey, measured as survival and growth rates. And what these experiments have uniformly shown is that there's always a really dramatic effect on growth of predation risk. There isn't always a dramatic effect on survival rate, because once they make the dramatic behavioral adjustment that results in poor feeding conditions, spatially restricted foraging, that very often prevents mortality rates from increasing as predator densities are increased in the experiments. Oh, there you go. So there's an emerging ecology of fear literature bandwagon in general ecology today. It all basically was predicted by this work.
Carl, any parallel to how we're behaving during COVID?
Yeah, we're down in there. Yeah, we're not doing a lot of things that we'd love to do, except that what's happening to us is we're getting fatter when there's more COVID around because we're hiding more and we eat more when we hide. But that we don't have to go out to get our food, right? There you go. Yeah, let me show you just a kind of simple example of this. So we used to do these predator-prey experiments where we put prey and predators in a vat, in an experimental vat. Like we'd put a large trout in there and a bunch of small trout, and we'd try to measure how increasing the prey density affected the number of prey eaten by each predator in the experiments. And if you're careful with the setup of the experiment, what you get when you vary the density of prey from low to high is you get what's called a type 2 functional response. Increasing the prey density, the predators catch more of them until you reach a point where the predator just can't eat anymore, or spends all of his time handling the prey that's already eaten. And so this is the predator satiates as prey density goes up in these experiments. But if you do the same experiment, but you just put some rocks in the aquarium, the pattern you see in the experiment changes completely, because the first thing that happens when you put the predator in is that the prey all run over here and hide behind the rocks. Fair enough. And when prey density is really low, they never come out from behind the rocks. So the predators don't get to eat any of them. It's only when you put in a high prey density that these little guys are too crowded in there and they get hungry and they come out to feed, and wham, they get nailed by the predators. So what you see is the predator's response goes up dramatically. This is called a type 3 functional response, and in ecological theory, this is known to have strong stabilizing effects on populations. It causes positively density dependent mortality rate in prey populations, which is an ecologically stabilizing influence. Okay. Yeah, don't need to go. There's just more details about how foraging arena structures develop in space and time. Now, we've been talking here about basically arena dynamics that go on down at spatial scales of meters or smaller even, and very short time scales of minutes to hours. So immediately the question arises: how can we calculate the implications of these fine scale dynamics on larger scale processes, particularly on recruitment patterns in populations that influence overall population dynamics, and on trophic interaction dynamics more broadly in general, that are unfolding at much larger spatial scales of landscapes and over much longer time scales of years to decades. How can we bridge this gap in scales between what we can see going on experimentally and behaviorally down at fine scales and what we see on other scales? One of the first success stories we had at bridging that gap was in explaining what are called Berton-Holt recruitment patterns. In the fisheries literature, we've collected data for over 300 fish populations where we've measured in various ways the abundance of spawning fish or eggs deposited in different years, and then how many recruits resulted from those eggs. And what we see in almost all of those 300 data sets is kind of a shotgun scatter, and it scares biologists because there's a lot of variability. But it isn't the variability that's important here. What's important is the shotgun scatter doesn't have a positive slope. That is, we see almost the same number of recruits when there are many fewer eggs as we do when there are a lot more eggs. So what's important about this is that when you have fewer eggs, if you're going to get the same number of recruits, then those eggs have to have a lot better survival rate. So what's striking in these data sets here is not that there's variability around the curve. It's that there's this really strong density dependence going on. So that reducing the egg production by a huge amount has relatively little effect on the number of surviving juveniles. And this is completely invariant to biology in terms of taxonomy or life history types. So shad are an anadromous fish. This is data from the shad from, I think it's the big river that flows through New York City, the Hudson River. This is some anchovy data from California. This is the northern cod population off Newfoundland. This is pollock from an area of the North Sea. This is walleye from a lake called Escanaba in Wisconsin in the US. And this is data from the southern bluefin tuna off the coast of Australia and South Africa. So wildly varying biologies producing essentially the same basic recruitment pattern. So what we did is we started applying the foraging arena theory to this, and it turns out that if you start out with the foraging arena idea that as juvenile density goes up, food density should decrease hyperbolically. We have strong empirical support for this from experiments and so on. It turns out that the amount of time spent foraging and active and exposed to mortality, in order to achieve a given growth rate, if food density is going down like this, it turns out another counterintuitive prediction. The mortality rate of the juveniles should go up linearly with juvenile density, right? If food density is going down, juveniles want to achieve the same amount of growth, or try to achieve the same amount of growth. It'll mean the amount of time they'd have to spend feeding has to go up as a straight line. And it turns out that mortality rate going up as a straight line with density is the assumption that was used by Brereton and Holt in their work in the 1950s to derive the thing called the Brereton-Holt recruit relationship between the initial juvenile density and the net number of recruitments of juveniles surviving through their first year or two of life. There's massive empirical support for this, for the occurrence of these Brereton-Holt relationships. So we're just saying everywhere you see it, you're seeing juveniles that are foraging in spatially restricted arenas and are seeing this hyperbolic effect of competitor density. That is driving them to have to spend, when they're more dense, more time foraging in order to get enough food to grow and survive at all. And that in turn is producing the larger scale Brereton-Holt overall survival pattern over the longer time scale of a year or more. So we figured this stuff out, I guess back in sometime around the early 1990s, mid 1990s. I'm not going to go through this. You can go through it. One of the predictions that we made right away is that when you increase predation risk, the relative recruitment rate should be very sensitive to that predation risk. It shouldn't take much change in predator numbers to generate a large change in relative recruitment in some populations. We had noticed that there'd been decreasing survival rates in coho and Chinook juvenile salmon out in the Georgia Strait over time, coincident with increases in abundances of some predators, marine mammals particularly, but also coincident with increases in number of juveniles because of stocking from hatcheries. So when we were building models of what had happened, we were building these models that said when you add hatchery production and put more juveniles into the Georgia Strait ecosystem, the wild production should stay the same, but you should end up with an increment and a lot more fish out there than you had before. But when there's foraging arena competition effects between the juveniles of hatchery and wild type in restricted spatial arenas and or increasing predation risk, then what we actually get out there is that increasing the stocking didn't increase abundance at all. In fact, the abundance stayed the same or even declined, which is the foraging arena type prediction of that large scale experimental manipulation of abundances. Another thing that is that the foraging arena equation predicts as you increase predator abundances, the predation mortality rate, instead of going up like a straight line with increasing predators, should go up and then bend over to a maximum rate set by how fast juveniles make themselves available to predation. So around this time, we were starting to sort these things out, and I happened to, another lucky accident, is I happened to walk through a room in our old hut where we, in our old Institute of Animal Resource Ecology or Fisheries Center hut, and Daniel Pauly and Billy were given a workshop on Ecopath, which is a static mass balance thing like that. And they were standing up there, I think Daniel particularly was waving his arms, showing this static data that had gone into the Ecopath model. And he was making dynamic predictions from it about how things would change if you changed the abundances of creatures in the system. And I just stopped and I said, that's just nonsense. You cannot take a static mass balance flow picture given flow rates at some equilibrium and predict what's going to happen when a system is moved away from the equilibrium just by waving your arms like that. And they agreed and they said, okay, fix it. Build a dynamic model for us. So I first sat down and I built one of these old simple mass action type models like I'd been building back in the 70s and stuck data from Ecopath into it as to initialize it and to calculate the trophic interaction rates, mass action rates. And just like them old 1970s models, the thing fell apart and didn't make useful predictions at all. And then it dawned on me, maybe this foraging arena idea, what if I model each of the trophic links in their Ecopath model with one of these vulnerability exchange equations that limits predation rates? And so I actually was in a spreadsheet model for a bay in the Philippines for Alida Bundy, who some of you know. I built this spreadsheet version of a predator-prey multi-species interaction model where I just put in the vulnerability exchange dynamics equations that we derived earlier for study and recruitment. And bam, this spreadsheet all of a sudden behaved. The system stopped exploding. All the species that were in it coexisted. It responded sensibly to changes in things like fishing mortality rates. And thus Ecosim, the Ecosim part of Ecopath was born out of that lucky accident. Do you want to add anything to that point, base point? Anyway, this was kind of cool. This was heady times. So Billy and I got together, and Billy said, "Okay, can we program this into the Ecopath software?" So we started jointly. I would write these bits of mathematical code to integrate differential equations and solve the foraging arena equations and that sort of thing, and I'd write them in BASIC and I'd pass them along to Billy, and Billy would stick them into the Ecopath code, and the code got progressively more complex as we thought to add more and more different kinds of clever things to it. And eventually, the emergent result after many years and many reprogrammings and so on is the software that you guys are going to be working with in class. So, just before I go on here, this idea of a vulnerability exchange process also applies to things like sport fishing. You know, you're sitting out in the middle of a lake and you've already cleaned out all the vulnerable fish because there's a whole bunch of fishermen out there, and the fish that are left are the ones that are reasonably smart. And so catch rates when the number of fishermen in a sport fishery goes up, or commercial fishermen even, catch rates go down much faster than fish abundances do. I was once involved in a really neat experiment on this out on a place called Bolt Reef on the Great Barrier Reef in the Capricorn Bunker Group near Heron Island, where a sport fishing club came out and fished the dickens out of a coral reef for 1 week. Everybody fished every day. Typical Australian bunch of drunks out there camped in their boats. Meanwhile, a bunch of biologists did dive surveys across the reef and measured how many fish were actually left. And it turned out that the sport catch per unit effort dropped to 0 by 5 days into the experiment. But the dive surveys showed that 80% of the coral reef fish that they were pursuing were still there. In other words, there'd been a huge impact of the localized competition interaction in those fishermen's foraging arena, driving down vulnerable densities when the overall abundances hadn't changed at all. they were not depleting the coral reef population because of the behavior of the fish was limiting the ability of fishermen as predators to deplete them. Fair enough. So you all know about Ecopath, Ecosim. I don't have to tell you. There's a, you basically build an Ecopath model where you specify base biomass levels, and then you use diet compositions and feeding rates to try to calculate baseline trophic flows between those along those mass links. So all we did with Ecosim was we put in used Ecopath to initialize the biomasses in these pools. And then for each of the Ecopath trophic links, we build an equation using the foraging arena theory for predicting how the trophic flow rate along the link should vary with the abundance of prey and abundance of predators, assuming that the prey exhibit behaviors that limit their vulnerability to the predators, and or the predators exhibit behavior that limit the vulnerability of the prey to them. So we went out and tested Ecosim on a really wide variety of ecosystems, mostly marine ecosystems, all around the world, mostly ones Billy had been working on. And this is the kind of thing that happens. So this is a model of the Georgia Strait, one of the first ones that we developed. This is the way that model behaves over time if you put in classic assumptions about mass action interaction and type 2 functional responses. It goes violently unstable, and within about 20 years of simulation time has gotten down to, from something like 50 functional groups, there's about 5 functional groups left. Just putting in the foraging arena structure, just limiting the trophic interaction rates using the vulnerability exchange equations, and bingo, the thing behaves quite sensibly over time. Now, the behavior here is complicated because we're also fitting that model to time varying productivity indicators and time varying fishing mortality rates and other things. But you see the dramatic difference that using the foraging arena theory has on the way a dynamic model of an ecosystem behaves. This is the most vivid example I can think of that. So, you know, we started figuring out how to fit these models to data, as I think you'll be learning in the class, and discovered that they're pretty remarkable sometimes at being able to explain all sorts of changes in abundances of creatures as being partly due to the way we manage those creatures, like releasing marine mammals from harvest control programs and so on. A lot of these effects are mainly due to fishing changes in fisheries. Some are due to trophic interaction effects. Here's a kind of important thing. When you're building one of these models, one of the first models we built was to duplicate Polavina's work on French frigate shoals in the Northwest Hawaiian Islands. The original Ecopath model, first one, was built for French frigate shoals. There was an interesting issue on French frigate shoals. There's been a decline in an endangered species, the monk seal, and it was argued that this was caused by a lobster fishery that went in and fished down lobsters on the French frigate shoals. There was a period of very high fishing effort, and lobsters had been depleted. So people were arguing that monk seals had declined because of the decline in lobsters. Well, monk seals do eat lobsters, but they eat relatively few of them, and they certainly don't depend on them. They have a lot of alternative prey types. So when we tried to fit the Ecosim model, it just would never predict the decline in monk seal abundance that one would predict if they were if lobsters were that important to them, and from the decline in lobster data. Also, the lobster population didn't recover in the system, and then these models kept wanting to have the lobsters come back when the fishing effort went down. Well, Polavina looked at this and he said, you know, there was a period of low chlorophyll in the ocean off the Northwest Hawaiian Islands for a long period here during the 1990s. Why don't you drive your model with that basic change in ecosystem productivity that we know occurred from field measurements of chlorophyll concentration in the Northwest Hawaiian Islands. Well, bam, we put in that chlorophyll data as a driver for the model, and instantly the model fit the data and also correctly predicted the failure of lobster populations to recover and the failure of monk seal populations to recover. But it said that this has not happened because of the lobster fishery. It's the productivity change that's changing the abundances of all of the major forage creatures for monk seals that are driving this dynamics out there, not that one fishery that people were trying to blame for the problem. So it's just showing how you a lot of times you're really learning the most from a model when it isn't fitting, when it can explain part of what you see, but it fails to explain other parts of what you see. And those of you who do follow up on modeling enough to actually do some, try to do some model filling, we'll see this in spades. You'll see your models failing, and then you realize, well, I left out some basic thing that I already knew was going on, like chlorophyll changes or a fishery or whatever. And I put that in, and the model works better. So, we went through and fitted models. You can look at this on your own time. All sorts of things. One interesting thing we discovered in places like the Great Lakes is that you can end up with an Ecosim model that works really well for some of the species in the system, but fails completely to explain patterns in other species in the system. Right? So when somebody says, is your model right or wrong, does it work or not work, there isn't a single answer to that. You can capture the things that influence the dynamics of some species quite well, while utterly failing to capture things that influence the dynamics of other species in that same ecosystem. Blah, blah, it goes on and on like this. Yeah, bottom line here, should we trust these predictions? Of course not, nor should you implement policies that will only work if some particular model is correct. But at least once you get one of these models to fit, you find an explanation that's consistent with historical data. At least you have a better chance of directing policy choices than the kind of boneheaded arguments that are now being used to justify policies that ignore interaction effects, like saying the mammals will simply find other food sources, or fishing at less than the natural mortality rate should not impair the ability of a species. The kind of simple-minded rules that we use when we don't have the models. Surely we can do better than that to the ecosystem modeling exercises. Okay, I want to close this part of the discussion and take a little bit of a break so that I can have a weed and go to the potty and answer a few questions. This is just a warning to you, right, about how wrong you can be. So here's Hilborn and I in our fisheries ecology textbook in 1992. There was a quote from us saying, "We believe the food web modeling approach is hopeless as an aid to formulating management advice. The number of parameters and assumptions required are enormous." Fair enough. Well, what did I just tell you? Hilborn and I one of the things you never want to do as a scientist is to rule something out without having tried it. And that's what Ray and I did back then. And it really took the combination of data assembly characteristics of the Ecopath approach and other modern approaches for pulling data together, along with improving model structure that made this thing we said back then a really very, very wrong. Okay, so that's the end of the basic foraging arena lecture. And let's stop at this point and have a little discussion here.
Thank you very much, Coll. It's a great lecture, and I think it's interesting that you end up with this quote and by saying that you were not right. And I think that's one of the things I really appreciate working with you, that you're willing to admit if whenever there is something that's not wrong, not gets hung up with it.
Mate, that's called science. If you got a scientist who never admits their mistakes, what you got is not a scientist. You got a Donald Trump. with all of the
Yes.
horrors that that kind of faith-based approach to living implies. You know, so you see
It's
a
true.
scientist, there are lots of scientists who don't admit their mistakes. And to me, those they cling to their beliefs no matter what the evidence might be around them. And to me, those people are absolutely no different from Trump. And they'll have the
And
same destructive influence that he has.
yeah, another thing I remember on this is when you talked about the example with Polovina and the impact of lower phytoplankton in the area, that your immediate reaction was, "Nah, that's not going to be it." But trying it is what we need to do. Test the hypothesis and see where that leads us. And is it possible to explain it with this? And yeah, it changed our view
Yeah,
on a number of
luckily,
things.
what had happened there was that Polovina was, we were in a workshop in Hawaii when this happened, and Polovina was in looking at these Ecosim runs that we were making and saying, that doesn't look right, that doesn't look right, and so on. And when he suggested that chlorophyll thing, one of the really important features we built into Ecosim is the sketchpad feature. So you can have these time forcing functions, and you can just go with your mouse and sketch a pattern without worrying about quantifying that pattern really precisely. So I just did it for Jeff. I just sketched this pattern in. It took me like 5 seconds to sketch a time forcing pattern for primary production where I doubled it or halved it for a period of time in the model, and then made the run within 2 or 3 seconds. You know, I don't know if you heard me. You heard me give a seminar a while ago about. Oh no, you wouldn't have. It was in a different kind of meeting. Oh, it was a meeting where somebody brought up what they called the toy models. To me, which to me is one of the most disgusting misrepresentations of modeling that you can possibly have. That he said maybe we should build a toy model to show people these ideas, and I said, what do you mean a toy model? Because every model is a toy. Every model is something you play with to try to learn things about the world and about at least how you think about the world. There's no, all models are toy models. To refer to a model as a toy model is to imply that there's something kind of model you could build that is not a toy, that would actually be the right model and incorporate reality correctly. That's a mistake that a number of ecosystem modeling teams are making. They're building these monstrous models that they can't play with, that they can't treat like toys. And I assert we can learn nothing from them. Fair enough?
Yeah. So Carl,
Play is critical. Never be afraid to change things and play with them. Play with your ideas. Learn from your mistakes.
why did the monk seal then decrease if it wasn't? Was it just prime production?
Well, yeah, we didn't follow up on this, but I gather that reef surveys in the area done by guys like Polovina and Evan Howell and so on in more recent years have supported this notion that there's been a general decrease in the abundance of all sorts of small planktivorous fish in that system. When they first went out and started looking at French frigate shoals, they saw relatively low abundances of planktivorous fish in the first place because the system was dominated by really large Alua. What are the big, large snapper, Coran jack species that just patrolled the reef edge and drove all the juvenile fish into hiding all the time and made it hard to monitor and also and so on. But the Alua population dropped pretty dramatically. But that didn't allow those juvenile the planktivores to build up in abundance. They stayed low. So there was something else that was preventing them. It was plankton and food availability. You know, some
Okay.
people don't realize about a coral reef, but most of the production that of fish on a coral reef isn't due to any production that goes on in the coral reef. Most coral reef fish biomass is of planktivores. About 80% of the biomass of fish on a typical reef are planktivores. Oh, so I guess herbivores can be pretty big on some reefs as well. And the planktivores are feeding on the plankton that's being produced elsewhere in the ocean and is being carried past the reef by currents. You see that if you go out and look at reefs, you see where are the planktivore densities highest? Well, they're on the upcurrent side, not on the downstream side of the reefs. That's where all the food is, on the front of the reefs. Okay.
All right. Yeah, thank you very much. Just the last thing about Carl's talk here is that it's a classic talk, and it was made by a time when he was trying to find the ugliest background for his presentation, and this is the winner of it.
Yay! Yeah, that was no. I did find one uglier color, which is pea soup green, but I had to change that. That was just awful.
Okay, if you would, Carl, if you would just stop sharing your screen, then I'll see if I can overrule him. I can hear. So the next thing is we're going to have some brief introduction, and Sylvia, I think you were going to start off. See, now I'm sharing and now I'm stopping to share. So, oh. Are we still?
Can you still see Carl's screen?
Could you, yeah, are we still looking at that now? Sylvia, if you would share your screen, or if you want to do you want to show something?
Yeah. Do you want to wait until Carl's back?
No, I think you could, if you go share screen, I think you should be able to take over. There we are. It works. Yes, where is that play button?
Sorry. So do you want me to start the...
Down there, yeah.
Okay. All right. I guess Carl is not here, but I was going to thank him actually for the great presentation. He really explained this in such great layman terms that it's really hard to to go close to that. But I will do a summary of his paper, I guess, on the theory that presents the theory and the challenges of the foraging arena theory, along with some very good examples. I'm not going to go over the examples. Obviously, there's no time for the one slide. So I'll just go over some of the highlights. So Ecosim is built around the foraging arena theory, where the assumption is that trophic interactions are largely limited to spatially restricted foraging arenas as a way for species to limit the risk for predation. So the prey can be either vulnerable to predation or invulnerable in this case. In other words, the theory proposes a way to define what is limited food supply for ecosystem modeling context. Spatial and temporal restrictions in predator and or prey activity cause the prey populations to partition into vulnerable and invulnerable population components so that the predation rates are actually dependent and or are limited by the exchange rates between the two prey components, the vulnerable versus invulnerable. So there are actually two basic models that predict the vulnerability state change for the prey. First is the continuous exchange model, where the change occurs constantly. And the second one is the feeding bounce model, where the change occurs only during dusk and dawn, when feeding mostly occurs as well. So the estimation actually of this vulnerability exchange rates has had a very limited success, considering they're very near impossible to observe directly in the field. Another thing that's actually good to remember for the foraging arena models that they assert is competition between predators is intensified from the spatial restriction of the interactions that occur within these foraging arenas. So based on vulnerability on or being non-vulnerable. Also is that the same behaviors that are used to acquire resources cause some of the species to actually become resources for other species, which is essentially at the basis of the predator-prey interactions. The predator-prey interactions on the other hand occur through behavioral and physical mechanisms, so that vulnerability can actually be driven by the need for food, dispersal, territory defense or aggressive behavior, accessibility or digestibility of resources. Also, vulnerability can also be modified by physical processes. For example, transport of nutrients, and Carol mentioned that, or strength of currents, for example, is another one. In addition, vulnerability can occur at a variety of temporal scales, for example, minutes to decades, or functional scales from individuals to an ecosystem level. Some of the major challenges for the practical application of the foraging arena theory in models are actually estimating the vulnerability exchange rates, which I mentioned earlier, predator rates of effective search, and change of these rates in areas with major fishing operations, which is it's very important when you're actually looking at managed systems. Why do we really care, and why is this theory so important? Because, as I mentioned earlier, UE is actually built around it. So more particularly, the foraging arena theory is one component that adds stability to the predator-prey interactions in the UE models. And finally, something that is actually really good to know is that these predictions from the foraging arena theory have actually been widely used by fisheries scientists to explain and model responses of harvested ecosystems, but they haven't yet been recognized widely recognized to understand or to model wide range of other behaviors. So it's something that still needs to be done potentially in the future. And that's it.
Thank you very much, Emil. And next would be the two, this is a more difficult task, these two Cecilia's, the two Ecosim papers.
Yes. So I reviewed the two Ecosim papers. So here we go. Okay. So there was two Ecosim papers that we were assigned as readings for this class. The first was the structuring dynamic models of exploited ecosystems from trophic mass-based assessments that Walters et al. Wrote in 1997, and another one from 2000. And I'm just going to really briefly introduce both of them. They're both fairly dense in terms of the mathematics, and I'm not really going to get too much into that. But basically, both of the papers present an introduction to Ecosim to convert the static Ecopath model into a dynamic one. And this is done through re-expressing the master equations of Ecopath as differential equations to account for changes in production, biomass, and consumption over time. So you can also add temperature profiles that change dynamically with seasons as well. And it also provides a way to deal with trophic ontogeny, where cohorts, like a juvenile cohort that has a different diet to its adult cohort, might be transitioning between groups, and it divides these groups into subgroups using Beverton-Holt factors, which Carl was talking about in his presentation. And so in the second paper that we would have read this week, Walters et al. 2000. This presents an improvement on the original Ecosim that they produced. Ecosim allows for the investigation of how biomass and other ecosystem flows change in a dynamic system, which is amazing because, as we all know, ecosystems aren't static, they are dynamic. And so if we have a way of actually looking at ecosystems in a more dynamic way, then we can model those changes and see how ecosystems might respond to changes. And it allows us to kind of investigate hypotheses about top-down and bottom-up controls of trophic structure and abundance. And so some limitations, as in Ecopath, is it doesn't explicitly model mesoscale spatial and temporal processes, for example, seasonal timescales or spatial scales. There is an assumption of additivity and mortality, which is not necessarily held up in real ecosystems. And a couple of other ones were the overestimation of potential productivity for low fecundity species, as well as overly optimistic assessments of Fmax for intermediate trophic levels, and indeterminate outcomes in complex food webs. And so that was kind of what I pulled out of those papers. Without, there's a lot, and then all of the equations are included in the papers as well, which I decided not really to get into because it just becomes a bit more, it's a lot harder than to describe. So I tried to float the main points.
Okay, thank you very much, Cecilia, and thank you, Sylvia, for two excellent introductions. And Carl, should we take 15 minutes of questions before we take the second round?
Why don't I do the second round and then do the questions? Let me just make one comment on that last presentation. Thanks for doing that one.
Yeah.
One of the first points in there was that the statement that Ecosim re-expresses the Ecopath master equations as dynamic equations. I've seen that appear in about 3 papers, and actually even with my name on it. That is not true. Ecosim is an ecosystem dynamic model that's actually completely independent of Ecopath. Ecopath only provides a convenient way to initialize the biomasses and abundances in the Ecosim dynamic model. But you can run the Ecosim dynamic model without ever having anything to do with Ecopath. Ecopath just helps in the initialization of the dynamic model and in calculation of some of the dynamic model parameters. But it's not just the reprogramming of the Ecopath master equation or anything like that. There's much more to it. Anyway, yeah, don't worry about that. That's no big deal. You can get away with saying that about the master equation if you want in the paper.
It's the components of the equations are carried over. I think that's one aspect of it.
Yeah.
It's not like Ecopath.
They're not carried over. Anyway,
it's the same components. You have the components in there, so.
Yeah.
I think it's a legitimate way of thinking of it, that there is that relationship. Okay, Carl, you wanted to continue with?
Yeah, I just wanted to take about 20 minutes to talk about 3 particular aspects of Ecosim dynamics that you really need to be careful with if you do build Ecosim models. 3 ways basically to make those models deal more realistically with certain kinds of issues that have arisen in ecosystem management. Why don't I do that and then we'll do the question period? Do you mind, or do you?
No.
If there's a really pressing question, we could do that, but otherwise, I'd rather.
I think we can wait 20 minutes.
What do you want to do then?
Go ahead, Carl. Go ahead.
Okay. Okay, we're sharing my screen and you got a picture of one of my grandkids. No, I think that's a picture of Will actually. Anyway.
That's kind of difficult to see.
Doesn't matter, it's all covered up. Alrighty, I'm going up to here and I need to get that laser pointer working like that. Okay. So I want to talk about three things that we've discovered working with Ecosim models that you've really got to watch out for when you're doing the model formulation. I'm going to talk about what's called trophic triangles and cultivation depensation effects that can lead to multiple equilibria in the dynamics. I'm going to talk about what the problem called non-additive predation rates that can make a model make incorrect predictions about what happens when you change predator abundances in a system. And then I want to talk about another mechanism that can cause multiple equilibria in a model, which is depensatory mortality effects. Now, all of these are related to the idea that if you fire up an Ecosim model, the default parameter configurations that are provided with the software will cause the model to exhibit what's called global stability. That means that if you disturb the biomasses by putting in a period of fishing and so on, and then you remove that disturbance, no matter how severe you make the disturbance, the biomasses will all eventually return to something close to the ecopath equilibrium. Fair enough. So the ecopath biomasses are assumed to be a global equilibrium in the way the parameters are set. But we know that that's not the way some ecosystems behave. We see ecosystems in some cases exhibiting what Halling called multiple equilibria or multiple stable states, such that if they get disturbed very far away from an initial or a balanced natural state, they will move to and persistently remain quite different than they were. The possibility of multiple equilibria is one of the most serious policy issues in ecosystem management. Is there a domain within which you have to manage the system without having it flip into some entirely different system configuration that may be quite undesirable and remain in that undesirable configuration no matter what you then do to try to recover it? Okay, well, trophic triangles first came up in models of the central North Pacific ecosystem. And then we realized that it applied much more broadly and more particularly to a couple of Canadian situations. So the European scientists have called this trophic triangles. I call it cultivation depensation. The idea is if you got a model, an Ecosim model that has a multi-stanza representation of a population, let's say cod, off Newfoundland. And so you have adult cod and the biomass of them, and then you have, and those adults are producing juvenile cod. Fair enough. And normally, if you just had this as a population dynamics interaction, you'd see limited recruitment of juveniles because of foraging arena effects, and you'd see a stable cod population. But in off the coast of Newfoundland, and there are also small species, including a smaller cod called the Arctic cod, some small benthic piscivores, and even herring that all prey when opportunity presents itself on juvenile cod, either on the cod larvae or on small cod juveniles. So what we think is going on in those systems is that when cod are abundant, as they were historically, they suppress at least behaviorally, or suppress the biomass of a lot of these smaller species. That reduces predation on the cod juveniles, which helps to maintain the large cod population. So we say the large cod cultivates the better ecosystem condition for its juveniles by keeping other species down. Well, the danger there is that if fisheries, say, reduce the cod abundance, then these other species may build up, knock down the juvenile survival of cod, and the system may move to a balance, a new equilibrium point that has only very low cod biomasses persisting for a long time. We see something like this off of Canada, off the east coast of Canada has been observed, where the systems have changed from what they call bentivore or piscivore or cod dominated to pelagic or small species dominated. And they persisted in these relatively unhealthy states from a standpoint of fisheries management for fairly long periods of time. Fortunately, the cod thing is not persisting in the planktivore dominated state. What will happen when you set up an Ecosim model that has this cultivation depensation structure is that the initial Ecopath equilibrium point, instead of being a global equilibrium point, it actually becomes a saddle point. Hang on just a second here. I've got to get my wife to... Sandra, the oven's going. My wife is making my weekly bread and the oven is dinging, telling her she needs to turn on the heat or turn it off or something. I'm not sure which. Sorry about that. There you go. Okay, fair enough. Where was I? Yeah, so thing is, watch out for it in the models. What we saw in the Central North Pacific was that when we knocked down, started the Ecopath base, and then we fished down some of the large pelagic species, we got explosions in some smaller pelagic species like mahi-mahi, and those high mahi-mahi abundances resulted in high predation rates on the juveniles of the large pelagic tunas, and the tunas crashed in the model. Fair enough. So watch out for it. It's an interesting dynamics, but be aware that in order to see it, you have to be careful about the Ecosim parameter settings. The default settings won't produce this kind of dangerous situation. The second one is
Coll, bass and bluegill lakes.
Oh yeah, there's a famous example of this called balance in farm ponds, where people have tried to manage farm ponds in the southern United States as a predator-prey system with a large centrarchid species called a bass and a smaller prey species called the bluegill. And they have a terrible time trying to make these farm ponds remain stable, and particularly keeping the bass around. Because what happens is that when bass are abundant, they eat the bluegill and keep the bluegill down. But when when bass get fished down, the bluegills build up, and then the bluegills eat all the bass eggs. These are nesting species, and the bluegills will sneak in and eat all of the bass eggs out of the bass nests. That's a famous example of this two equilibrium management problem.
And very remarkably, we can actually replicate that in a simple model that it seems to work.
Yeah.
Get it to shift from one to the other just by a bit of fishing.
There's an American university called Auburn University, where famous people who studied farm pond biology did hundreds of experiments stocking bass and bluegill at different densities and then monitoring what happened over time in them and then draining the ponds so they got exact numbers. Someday somebody's going to get all of that, what's called the Swingle-Smith database, out of Auburn University and do some really neat analyses of this whole business of cultivation depensation in an experimental setting. The data are there somewhere, just a matter of getting them out. Okay, the second thing is that it's possible for small species to have high mortality rates, whether or not predation rates are high. That is, reducing predation may not make small species more productive. In simple food chain like Ecosim models, you get this kind of trade-off. If you take the large piscivores down by fishing, you get increased productivity of small species, small planktivores in particular. So if you were trying to manage an ecosystem to just produce protein, fish protein, the models that are like that will tell you that the way to produce protein is to knock out all the competition, knock out all of the piscivores, and then just grow small species that grow fast and produce high biomasses. And if you want to feed the world, that's what you do, called balanced harvesting. Yeah, simplify the food web the same way we do in farming, for example. But the basic problem is that mortality rates of these small creatures may not be additive. And the way to see this is that it's quite possible that the vulnerability exchange process for a lot of these small fish may be a process that involves healthy fish becoming stressed because of factors like temperature and disease and catching diseases, from which they may or may not recover if a predator's not there. But if a predator is there, the predator may take those stressed fish. So it's a vulnerability exchange process, but it's one that's mediated through dynamics rather than just through simple behavioral exchange processes. When there is such a thing, when the vulnerability exchange between vulnerable and invulnerable creatures is mediated by a third thing like disease, changing the predator numbers may have very little effect, because it may be that those stressed fish would just die anyway. The other things would kill them because they're stressed. You don't know. You have to do an experiment to determine whether, if you reduce predators, those stressed fish would still die or would have instead recovered and contributed to productivity of the small fish population. Fair enough? Anyway, we've been looking at this in the Georgia Strait ecosystem. The simple models that you have mass action flow just don't account for this vulnerability exchange due to things like disease. If we assume in the Georgia Strait that the vulnerability exchange rate is somehow dependent on water temperature, we can calculate that how the vulnerability parameter of the exchange process has apparently increased in correlation with temperature changes. So it is apparent vulnerability is correlated with temperature. And our models then for predicting how, in this case, salmon populations, mortality rates in salmon populations will respond to changes in piscivore abundances in marine mammal due to marine mammal piscivory, change dramatically. When we assume additive predation, we predict that taking down the number of predators will greatly reduce the mortality rates of those juvenile salmon. But if we assume that the juvenile salmon are becoming vulnerable to the piscivore, to the marine mammals, because of something like disease or temperature, then when we reduce the predator population, we get very little change in the mortality rates of the juveniles. So the big management experiment in the Georgia Strait is clearly to reduce mammal abundances and to determine whether or not predation mortality rates really are additive. So that's the second one, is you got to watch out about complicated factors that generate complicated changes in these vulnerability exchange rates in the foraging arena equations. They can be driven by factors that we don't usually model in ecology, like disease and temperature changes. The third thing is a little more subtle here. When you have an efficient predator like a marine mammal off our coast here that has a large number of alternative prey sources, so that any one prey like herring that seals and sea lions like to eat, so the predator doesn't respond to changes in the numbers of that one prey type. That efficient predator may maintain high consumption rates of the one prey type, like herring, so that the predation mortality rate M, which is how much the predators eat over the prey biomass, will go up as prey biomass goes down. And this causes the prey to exhibit what's called depensatory declines due to increasing predation rates as prey biomass decreases, even if the predator biomass stays the same. If the predators then switch off when don't bother pursuing the prey when prey densities, when herring, if seals and sea lions don't bother chasing prey when herring are scarce, then the herring will have a chance to recover a little bit. And you get what's called a predator pit effect. And again, the herring biomass dynamics can have two equilibria. One at a high herring abundance and one at a low herring abundance, where every time the herrings start to recover, the marine mammals switch on again and knock that herring population back down. And in between these two stable points is an unstable point. Simple population models, when you look at productivity versus stock size, just say higher abundance should be less productive. With the depensatory predation, you get this much more complex structure. And when you plot the equilibria of herring against fishing mortality rate, if there's a lot of predators, the equilibria can have this funny looking curved pattern like this, with a high equilibrium and a low equilibrium. That pattern is called a fold catastrophe in the mathematical theory of dynamical systems, where there's lower fold is a stable point and the higher fold is a stable point, and in between is an unstable one. And what happens in this case is that it can be that once the stock of herring is collapsed to a low level, the only way to get it to recover is to greatly reduce predation mortality rates so that it can bounce back up. It also means that as you increase predator abundance, there's this kind of cliff edge, and the herring stock will just suddenly collapse on you out of nowhere if you're not closely monitoring the predation mortality rate as well as the fishing impact. Now, unfortunate. Yeah.
Carl, one more thing there. Well, for herring, it's also known to occasionally have good recruitment.
Right. Yeah.
Good recruitment can bring it over that cost again.
Yeah. One of the widest observations in the ecology of small planktivores in the oceans of the world has been that they tend to have these abundance regimes. They tend to be at high abundance for some time and low abundance for some time, and in high abundance and low abundance. And people have attributed this to changing recruitment regimes, the favorable regime and the unfavorable regime. Well, predation can generate the same effect of periods of high and low abundance, but with much more serious management implications, because of the possibility of controlling when the thing is and how it's going to flip between regimes. One of the problems in Ecosim is that very often predation mortality by marine mammals and large piscivores is seasonally concentrated on small planktivores during times when the planktivores are concentrated particularly for spawning. So herring are eaten off our coasts by marine mammals in huge tonnages, primarily during the herring spawning time, when the herring concentrate in small areas to spawn and are really, really available to the marine mammals to eat. Most of the year, the herring are spread out over wide areas of ocean, and the marine mammals don't bother chasing them because they're just too dilute. Ecosim doesn't have that kind of structure built into it. So Ecosim, unless you build it in, Ecosim assumes that when a predator like a seal is foraging its way through the world, it's encountering herring and other kinds of, and hake and other kinds of prey all the time as it's moving around. When you use that assumption and calculate how much herring it should eat as a function of herring biomass, you predict that it should go up smoothly. And they shouldn't eat much at low herring biomasses because they're eating a lot of other kinds of things at the same time. But if you assume that the herring consumption is that when the predator is feeding on herring seasonally, it's doing it over a short seasonal period, then the predator predation rate response, predator functional response is much steeper, and the compensatory predation effect is much stronger than the default assumption that you get with a standard Ecosim model that doesn't explicitly recognize that seasonality in targeting different prey types. So the advice here to you, if you get into Ecosim modeling that involves efficient predators like large piscivores and marine mammals, is watch out for strong seasonality in the vulnerability of particular small species to the predators, because that seasonality will cause the stability properties of the actual dynamics out there to be very different than a simple Ecosim model will predict when it's not accounting for the temporal organization of the interaction pattern. Fair enough?
Coll,
Okay, that finishes my little warnings to you
no,
about things that can go wrong. These are the biggest three things I've seen in all the experience I've had with Ecosim.
Coll, let me just ask one thing here on the left figure here. So in Ecosim, there is weight contraction with regards to fishing and fleets.
Mm-hmm.
Couldn't we have a similar concentration factor that takes care of this? You said you could
No.
build it in.
No. No.
Because it's seasonal?
Because it's a seasonal structure, yeah.
It's not just schools.
Yeah,
No.
it's the only way you can properly capture in an Ecosim model of anywhere along our coast the way marine mammals target herring is to have a herring spawning season when they eat primarily herring and exhibit a steep functional response to herring abundance, and then to have the rest of the year when they're targeting others or their foraging is spread out over much wider areas or they're targeting other prey types, but they are not targeting herring during those other times. Now, there may be some way to capture this without explicit seasonality, but I've never been able to think of it. There's a thing called essentially there's a switching function going on here. There's switching in Ecosim. You can turn on switching on the prey types, but that's not what's actually going on here. This is a more complex targeting process related to seasonal high densities of particular prey types. Seasonal high availability of particular prey types.
All right, we have 15 minutes left, so let's open it for questions.
Okay.
If you write it in the chat, then we'll take them as they come.
Do I have a chat here someplace?
Somewhere you do, but I don't know if I have any questions there. Don't quite believe that you were explained everything so clearly that there will be no questions.
Well, you know, if all this seemed really complicated to you, like talking about things going on scales of a meter in a few seconds and then trying to figure out how ecosystems behave, just remember this. An ecosystem is not a machine. You know, a lot of the modeling techniques that we developed in the early days were borrowed from engineering, where engineers were trying to describe how things like chemical reaction systems and machines work. Those are constructed systems that have a known set of components. And you know what all the pieces are, and you know how each of the pieces is supposed to behave. So you're supposed to be able to put the pieces together to troubleshoot for emergent problems that you wouldn't have thought of if you just looked at each piece, how each piece works. Ecosystems are not constructed. They are assembled through really complex evolutionary processes. And they can have these incredible complexities associated with the behavioral, particularly the behavioral ecology of the organisms. that you would never predict if you treated those organisms as machines. This is one of the reasons why whenever I hear the word physiological ecology, that's physiologists pretending to do ecology and making predictions about ecology using physiological principles like bioenergetics and growth principles. I just think that's just laughable. What really makes ecosystems complex is not just their incredible taxonomic diversity that develops over time, but also the functional behavioral complexity that allows that biological diversity to be there. All the little tiny ways that creatures use to avoid not getting wiped out by competition or predation and so on, to use space in all sorts of complicated and smart ways. So you never assume that just because you build a model with a bunch of boxes that you've captured that incredibly complex biology in a useful way.
Carl, there's a
Thank you,
really
Carl.
nice question. Oh, sorry, Billy. There's a
Go
really
ahead.
nice question
No, I was
in
just
the
going to do
chat.
exactly the same, yeah. And I was also
Okay.
going to ask Carl, you can stop sharing your screen perhaps so we can see some more people.
Oh, okay.
And Emma.
You're muted, Emma.
Should I read my question? Okay.
Go
I think
ahead,
I
please.
might have just been getting very confused, but I'll ask my question anyway. So my question is, if CPUE as a measure of abundance misses fish that are hiding, should we be inputting CPUE abundance estimates into models that are based on foraging arena theory like EWE? So in other words, if the model itself accounts for fish behavior, but some of the data we're feeding it does not, does that matter for the purposes of modeling?
Well, if you're talking about CPUE data like from a fishery, you're trying to use CPUE data as an abundance index. Is that what you're talking about? As data for testing the model? I have a very simple piece of advice is CPUE is never proportional to abundance. And you should never use it as a relative abundance index in any ecosystem modeling exercise if there's any way to avoid it. There's two things that can go wrong. The Bolt Reef example, what happened is CPUE dropped really rapidly when biomass out there didn't. So in that case, the fishery exhibited what's called hyperdepletion in the CPUE. CPUE went down much more rapidly than the biomass. This also happens in places like lobster fisheries where the lobster fishermen will go out at the start of the season and they'll fish on concentrations of lobsters or abalone, and they'll fish them down, and the CPUE drops very rapidly. But those concentrations are only a small part of the total population of lobsters or abalone, and so the overall population hasn't actually changed that much. So that's hyperdepletion. The other opposite situation occurs when you have aggregating prey species, and the prey keep re-aggregating as they're fished down. In that case, the fishermen see the same density of fish as the size of the fish population shrinks. That's what happened with cod. So the cod off Newfoundland, as their total abundance dropped, the area that they occupied shrunk down and down and down, but their densities within that occupied area stayed the same. So the fishermen's CPUE stayed the same. So the CPUE didn't drop when abundance dropped. That's called hyperstability. And that's actually the more common problem. It's more common for CPUE data to fail to reflect declines that are actually occurring than the other way around. So my advice to you real simply is don't use CPUE. Fishermen are too complex in their behaviors and fish are too complex in their behaviors to permit that kind of simple mass action assumption about how fish and fishermen interact to ever be valid.
Carl, if the alternative is
Thanks for
nothing,
bringing up that question, incidentally. That's a good one.
if the alternative is no data, can informed use of CPUE not?
Well, one thing you can do is to take the CPUE data and raise it to a power. If you raise it to a power more than 1, you're modeling hyperdepletion. If you raise it to a power less than 1, like 0.5, you're measuring hyperstability. So there's some empirical studies of how that power varies in different fisheries and for different survey methods and so on like that. So what you can do is you can run alternative Ecosim model fits. You can say, okay, I'm gonna use this index that's hyperstable, or I'm gonna use this assume the index is hyperdepleting, and then ask whether it makes a big difference to the parameter estimates that you get in Ecosim or to your conclusions about how things have changed. But that's the best you can do, unless you can get at the unless you can find out enough about the fishery to know how the fishermen behave, to have some idea of whether, and the fish behave, to know whether you're likely to be dealing with the hyperdepletion or hyperstable situation. Once you know the situation, yes, you can correct by just raising the CPUE to a power, but you got to know what power to use sensibly.
Thank you. Are there other questions?
Oh, the other one actually, the place that you got to watch out for is where there's been sequential depletion. So in abalone fisheries along the coast of Mexico, for example, there was a period when CPUEs remained stable for a long time and then they crashed. And what was going on there was that the abalone stock was crashing all the time. They were just moving further and further from port to fish each year and catching the same number of abalone, but just they were spending more time traveling to the fishing locations, but that wasn't reflected in the CPUE data.
Maybe following up on this, Carl, a question that relates to Andre Pont. You know, he's been saying never use output from one model as input for another model. Indirectly saying don't even think about making complex models of multi-species models rather than complex of ecosystems, because you won't be able to get the data for it, since you can't use output from assessment models.
No, that's just wrong. There's lots of times when assessment methods like stock reduction analysis and analysis of age composition data and so on allow us to get quite credible reconstructions of biomass changes over time that are not sensitive to problems with things like CPUE. And there's absolutely nothing wrong with using those biomass reconstructions as targets in CPUE, targets in Ecosim model fitting.
Good.
Okay. When you're doing the model fitting, what you're not trying to do is pretend that you know what's going on. You're saying that you have a time series of data that you think reflects the pattern of change that occurred in one component in the ecosystem. And then you ask whether or not the Ecosim model can reproduce that pattern of change you think took place for other reasons, whatever those other reasons may be, whether you got the pattern from a stock assessment model or a relative abundance time series or whatever. Stock assessment models are particularly useful because a lot of times you get relative abundance data for short periods of time, like from biomass surveys taken over short periods of time. But the stock assessment model can reconstruct biomass changes that likely occurred over longer periods of time by accounting for changes in catch and fishing impacts and that over time. And that's a quite credible reconstruction of how the population behaved. You know, it isn't perfect if that population actually changed in really complex ways due to trophic interactions that are seen in the Ecosim model, but were assumed away by assuming constant mortality rate in the stock assessment model. That would be wrong. But one of the things you can do is go back and forth. You can run a stock assessment model with constant mortality rates. Try to fit your Ecosim model to that, and if that Ecosim model tells you that the stock should have had quite variable mortality rates, you can take the Ecosim predicted mortality rates, plug them back into the stock assessment model, and update the stock assessment model with time varying mortalities. So you can iterate back and forth between the single species and multi-species calculations to learn more about both of those models.
Just has a question to follow up on this. Just, do you want to ask?
In the chat, or should I show it in the chat or something?
It's in the chat, yeah.
Who is this now? Just?
Yes, excuse me. My connection is not too good. So my question is if we could estimate biomass from CPUA using methods like that as CMS, right?
Yeah. Just as a warning, CMSY is a bogus methodology you should not use. That's based on particular very restrictive assumptions about surplus production dynamics that are violated in at least 50% of the world's fish population time series data. So do not use CMSY. When it's fitted to CPUE data, it isn't called CMSY. They have the people that do that have another name for it. It's a Bayesian surplus production fitting procedure. Those surplus production fitting procedures to very limited time series data or to assume depletion patterns in the case of CMSY can be really misleading. It is a way of doing what we call stock reduction analysis, but my recommendation is to start out with when you need to construct a time series of abundance estimates, start out with a sound population dynamics model in the first place that's structurally sound in terms of how long the creatures live and how they grow and how their recruitment is likely to have varied with stock size. And do the same analysis with that model that you could do with CPUE or CMSY while assuming a logistic growth model for the dynamics, because we know that logistic growth model is almost always going to be wrong.
It is a problem we often are facing because when we don't have information about biomass trends, it really means that there's very few constraints in our ecosystem models.
Yeah, that's right. Well, yeah, it just means there's very limited. Now, but before you go on here, let me recommend to you a paper that Billy can get for you that Billy and I wrote about. It was called Surplus Production Dynamics in Declining and Increasing Populations. And what it does is it's actually, there's been better versions of this done by Vertprey and Hilborn and others at the University of Washington. But there's a nice graph that Billy did in that paper where we took a whole bunch of the stock assessments around the world where they'd had pretty good reconstruction of abundances and surplus production patterns. And Billy just plotted surplus production versus biomass coming from those assessments. Now, if the logistic surplus production model held, we'd see in those data a kind of dome-shaped relationship. And what we see instead is in at least half of the data sets, we see clear temporal violations of any such stationarity assumption. We see changes in the apparent production relationship over time that are not, how much is produced at a given biomass changes systematically over time in at least half the data sets.
Now, I just gave everyone the wrong reference for it.
Shame on you.
It was another Hilborn paper.
What did you do?
Right one.
Cecilia has a question as well.
Yeah. Where am I looking?
Hi.
Oh, Cecilia, a lot of ecosystems have seasonal oscillations where interactions depend on seasonally abundant prey, like zooplankton bloom condition. Can that be incorporated into Ecosim? Is that your question?
Yeah, so kind of, it's more like if you're looking at the ecosystem over the course of an entire year and you're trying to create an Ecopath model to do that. I know Ecopath is static, but like, can you kind of incorporate those temporal variations into those models?
Our original aim when we were developing the Ecosim models is, and it was similar in developing Ecospace, the spatial dynamics models, was to look at longer-term changes on changes in multiple years over timescales of multiple years and decades. To do that, we integrate the dynamic equations on a much shorter time step of 1 month. Otherwise, they're numerically unstable. So that there is the structural capability in Ecosim and Ecospace for dealing with seasonality of the type you're talking about, like seasonal plankton production. But, you know, the way I warn people who are about to try to incorporate such things in an Ecosim model is that putting in seasonality is kind of like if you put it in one organism, like a zooplankton, that's kind of like saying you only want to be a little bit pregnant. You got me? Is that you put it in one part of the system, and then in order to get the story, you have to put it everywhere. So as soon as you make one part of the dynamic seasonal, it just forces you to a whole bunch of a huge explosion in assumptions about seasonality and all the other rate processes that are linked to it. Ecosim can often work pretty well by essentially treating everything as though it were not seasonal. So what it's doing is generating the integrated average annual effect of the seasonality over longer timescales. But when you try to model it explicitly, you can get into lots of trouble. The one exception to this where you really do need to include the seasonality is in this business of seasonal targeting by big efficient predators of multiple prey types. Generally, we've found that when you do, and it's easy to do this because you can put in seasonal time forcing on plankton productivity and so on very simply in Ecosim, is set up an Ecosim model with and without seasonality down at the bottom of the system. That's where it gets troublesome. It's with zooplankton seasonality. And what you'll see is that including that seasonality doesn't actually change the long-term dynamic predictions much at all. So things get better and things get worse, and the Ecosim model is kind of running through the average of them. And thank goodness that violent seasonality that we see in all sorts of processes like reproduction and availability of food and issues like match mismatching, prey and predator juvenile abundances gets smoothed out.
Thank you, and we
Good
have to
question
round off.
Anyway, yeah.
Yeah, we have to round off this this class here. It's past 11 here in Vancouver now. So I would like to thank you all, and especially, of course, I would like to thank Coll. And let me just mention that on Tuesday, Coll is going to come back, and he's going to be working in a spreadsheet demonstrating how to create a dynamic model. And
yeah, the idea there is that a lot of times students, if you haven't done it, it's not entirely clear what the logic is behind dynamic modeling and what's actually going on in a kind of philosophical or logical sense. So I'm going to build a series of very simple spreadsheets with you to make it abundantly clear just what these more complex codes like Ecosim are actually doing behind the scenes.
that's on Tuesday. So for now, thank you very much to you all, and especially Coll. Thank you.
Thank you very much.
Thank you.
Thank you so much.
Thank you so much.
Yeah, thank you so much, Coll.